Biology. Today, we are looking at DNA and RNA structure. This is Unit 6. Welcome to Unit 6. Yay! Unit 6 is my favorite. I get a kick out of this every time. DNA, RNA structure. This is topic 6.1. This is actually a review. And I don't know if they did this on purpose, but this is actually a review of Unit 1.6. So 6.1 and 1.6 are very similar. Anyway, let's get started. So DNA was discovered in this really cool way. So this is how it was discovered. In the 1950s, there was a woman, Rosalind Franklin, and she performed what's called X-ray crystallography on a piece of DNA. Essentially what she did was she shot a laser through a piece of DNA and it produced almost like this X-ray image. You can actually see it right down here. This is the real image of what she captured right down over there. Her work kind of revealed a pattern. Remember, all great scientists aren't scientists to begin with. They're just observers. They just see things and they thought it was weird. So she did this experiment. She looked at it. She thought it was weird. It was regular. It was repetitive. You can see sort of an X there and an X there, that sort of thing. So she kind of just sat on that information. She was like, this is weird. Turns out, and we know this now, is that what she was looking at was actually the major and minor grooves of the DNA structure. But she sort of sat on this discovery. She didn't quite know what was going on until that same time. So as Rosalind Franklin was doing her experiments, this other scientist, Edwin Chargaff, he analyzed different DNA samples from different species. So it took one from over here, one from over there, and he found, by looking at all of these different DNA from all of these different species, he found that one rule held true for everything. And it's a rule that you know really well. He found that the amount of adenine equals the amount of thymine in every sample. In every sample. Doesn't matter if it came from a plant or your cat or me or you. Every single time, the amount of adenine equals thymine. And he was like, oh, that's weird. Then he found that the amount, this is super weird, the amount of cytosine equals the amount of guanine every time in every species. Doesn't matter where it comes from. Isn't that wild? So he was like, this is weird. And Rosalind Franklin was sitting there being like, this is weird. They both had pieces of an answer, but they didn't quite know what was going on just yet. What Chargaff also discovered, actually, I don't think Chargaff discovered this, but what he was looking at was this idea of a purine and a pyrimidine. And this is a review of Unit 1.6. So we've seen this before. I'm actually just going to dump this on you there. Is that purines are these adenines and these guanines. And purines, do you remember me doing Beyonce in Unit 1? If it's a purine, then you're going to put a ring on it. So that is purines. It's got that extra ring on there. There's that double ring structure. Pyrimidines, single cut, right over here. So these single structure, single rings. So what we're looking at here is that purines bind with pyrimidines and vice versa. A purine's never going to bind with a purine. Their rings would get caught. I don't know. However you want to remember that. But if it's a purine, then you're going to put a ring on it. So what this amounts to is essentially this pairing idea, right? Is that adenine and thymine can kind of pair together, and they're held together by these hydrogen bonds. Remember, Unit 1, hydrogens make hydrogen bonds. There's a hydrogen, they make hydrogen bonds. It's like the one time chemistry makes sense. And this is sort of how they pair up, is that the ring of the purine is going to go over there, and it's going to align with this pyrimidine over here. And they see the same thing with the guanine and the cytosine. Pretty cool. Boom, hydrogen bonds. I highlighted them there for you in case the arrows weren't enough. Adenine and thymine have 2 hydrogen bonds, and cytosine and guanine have 3. We're going to see this because at some point during this lesson, we are going to build our own DNA molecules, and we're going to see that adenine and thymine make 2, and guanine and cytosine make 3. So think for a second, which one of these 2 pairings is stronger? I love hearing you think. Okay, let's do a quick review. Think ahead to DNA replication. How might hydrogen bonds aid in this process? Pause the video to think about it. Hydrogen bonds allow for DNA strands to be separated, right? Remember, hydrogen bonds are like the weaker of the bonds here. Together they are strong, but individually they are weak. So these hydrogen bonds allow for the strands to be separated. It's like a zipper. We're going to pull the zipper apart. A DNA sample is analyzed. We find that 22% is cytosine. How much thymine would we expect in this sample? Think of Chargraph's rule. Pause it here. Or think of my rule, that the curvy letters and the sharp letters. Pause the video to think. Jot down. Talk to each other. Yeah. 22% is cytosine. How much thymine would we expect to see in this sample? So cytosine is the C, and it would pair with the G. If you got 22 over here and 22 over here, that equals 44%. So 44% of this DNA would be guanine and cytosine. But they're asking about thymine. So what are we going to do? Well, if 44% is this pairing, we're going to take 100 minus 44, right? Which, you know how bad at math I am. $56. I just wanted to check. So 56% of it would be the other pairing. 56 divided by 2. Is 28. So 28% of it would be this, and 28% of it would be this. Remember to pay attention to what the question is asking. Great, I'm proud of you for getting that right. So let's move on. How did we discover this DNA? So what do? We got Chargraph over here saying A's pair with T's, and C's pair with G's, and then we got Rosalind Franklin over here being like, look at this weird structure, and along come these two dudes. Let's be real, white dudes. Watson and Crick. So he took the work that Rosalind Franklin did, and he took the work that Chargraph did, and they put it together. They did like a bunch of math. They were like, they were like math guys, to create this 3D double helix model of DNA. And it kind of was a solution to the questions that Franklin and Chargraph had. Like, this looks weird, and they kind of put it together. Franklin and Chargraph provided the evidence. Watson and Crick provided the reasoning. Let's look at what they found. So they found that DNA is a double-stranded helix. What does that mean? That means that it's got this backbone that's sugar, right? Backbone that's sugar. It's made up of sugar phosphate backbone. If I had a biology band, I would name it that. And in the center here, you got these nucleotide pairings. And it's double-stranded. There's one strand on top, and there's one strand on bottom. The strands are anti-parallel to each other. And what that meant was that there was directionality to it. If you recall from the mirror of this unit topic, right, there's a 3' end. When I said mirror, I meant like 1.6. There's a 3' end and a 5' end, and they're anti-parallel to each other, where the 5' ends are on the opposite sides and the 3' ends are on the opposite sides. This is really important when it comes to, when it comes to replication, because we need to know where to start and where to end. There's like directionality to it. So one of the strands runs from 5' to 3', and the other one runs from 3' to 5', if we're looking in that same like left to right direction. We can identify which end is which because your 5' end is always going to have a phosphate group, and your 3' end is always going to have a hydroxyl group. Remember, hydroxyl is the OH. Another Unit 1 throwback. So looking at it molecularly, we can figure out which way is which. Boom, right there. In case the arrows weren't enough, I highlighted it in red. Pause here if you got to take notes. Key functions of DNA. So DNA's got some key functions. As we know, it is the primary source of heritable information for every living organism. Every living organism. Genetic information is stored and passed from one generation to the next through DNA. So whether you are sexually reproducing organism, that you go through meiosis, and then come together through fertilization to create a new organism, or whether you are asexually reproducing going through mitosis. Either way, this information is passed from one cell to the next cell. The big exception to this, and I mean, it's kind of an exception, it's more of a caveat, is that RNA is the primary source of information for some viruses. As we know as scientists, RNA is still made up of those nucleotide base pairings. It's just a different kind of base pairing. We'll replace that thymine with that uracil, but the idea behind it is pretty much the same. Oh, and RNA is single-stranded. That's the other big difference. But it's still like AU, CG, you know? And it still passes down that heritable information. It's just not through DNA. It's through RNA. So pause here if you got to take notes. Love this prokaryotic versus eukaryotic DNA. I actually just went through this with my biomed class. So eukaryotic cells, as we know, they're more complicated as opposed to prokaryotic cells. Eukaryotic cells are larger. They have membrane-bound nuclei, whereas prokaryotic cells don't. Let's just bring our brains back to where those comparisons are right now. Remember, eukaryotic cells' DNA is found in the nucleus, and their chromosomes are linear, meaning there's a start point and an end point. They cross over like an X, like we can see this here in the centrosomes, right? That's sort of how they condense. But if we were to like pull it apart, it would like have one end that starts, and then it would like weave out, and one end that ends. In prokaryotic cells, as we know, they don't have like membrane-bound organelles. So they're hanging out in what we call that nucleoid region. And the chromosomes are circular, right? So there's not really like a start point and an end point. It kind of goes, it's packed in a similar way, right? So like it's condensed and that sort of thing. And if we pull it apart, it's like a circular sort of thing. In prokaryotes and some eukaryotes, they also contain plasmids. Plasmids are small little bits of DNA. They're circular over here, and they're separate from the chromosomes. If you can think back to the case study that we did with Dr. Bassler in the quorum sensing in bacteria, the signals that were sent out during quorum sensing was actually sent out from plasmids. In this unit, we're going to be working with E. coli bacteria, which is a prokaryotic cell. The DNA that we're going to insert into this E. coli is going to be a plasmid DNA. So plasmids are actually really beneficial for prokaryotic cells. We're not going to have to go into the chromosome to have to like replicate some of these genes. Pretty cool stuff. We'll get into it. This is kind of high-level overview. Pause here to take notes. Let's talk a little bit about plasmids. Yay! Plasmids replicate independently from the chromosomal DNA. So chromosomal DNA over here, hanging out in the nucleoid region, just being chill. And plasmid DNA is right over here. In this model, we're looking at 3 different plasmids, which is fine. There's some prokaryotes have multiple plasmids, primarily found in prokaryotes, and they contain genes that may be useful in a particular environment, but not required for survival. So I play a ton of video games. I'm a big gamer. This is like your quick thing on like your quick belt, right? On instead of going into your main menu to grab something, right? So if a change in an environment occurs, right, we're going to get that DNA from that plasmid. Plasmids can be manipulated in laboratories, so we're actually going to do this in our lab. They can be removed from bacteria, then a gene of interest is inserted into the plasmid to form recombinant plasmid DNA. When the recombinant plasmid is inserted back into the bacterial gene, it's going to be expressed. So we're actually going to do this. We're going to take a piece of DNA and we're going to insert a gene into it, and we're going to put that into E. coli, and the E. coli is going to be expressing that. It can exchange genes found in plasmids with neighboring bacteria. So actually, sorry, before we get into this, pause here to take notes. Awesome. Bacteria can exchange genes found in plasmids with neighboring bacteria. So this is Dr. Bassler's experiment that we did with the quorum sensing. So this is how, plasmids can really, or not plasmids, bacteria can really easily communicate with each other. They'll have these communication signals in the plasmids, and they'll be able to exchange it back and forth. So DNA can be exchanged through these plasmids. So it's like, hey, I got a gene that's helpful for our survival. Why don't you go ahead and have it over here, right? And they can kind of switch them back and forth. It's an evolutionary trait that they've developed over time. It's also, if we're taking a look at it, right, one of the reasons to support our endosymbiotic theory. Awesome. Take a look at this. RNA versus DNA. Isn't it beautiful? The only difference down here is this one. We got uracil. You're still looking at purines and pyrimidines. If it's purines, then you're going to put a ring on it. And pyrimidines down over here. RNA is single-stranded. DNA is double-stranded. So let's look. Let's look at some differences. RNA versus DNA. RNA have a ribonucleic acid. It's single-stranded. And A equals U, C equals G. It breaks my rule of base pairing when I say the curvy letters go together and the sharp letters go together, right? A goes to U, C goes to G. U, C, G are curvies. That's it. This is one of those rote memorization things. If you see a uracil at all, you know that we're talking about RNA. DNA is deoxyribonucleic acid, deoxy, so there's just extra oxygen in there. Or without, deoxy, I apologize, deoxy. DNA is double-stranded, and A pairs with T, C pairs with G, Chargaff's rule. My rule is the spiky letters go together and the curvy letters go together. So those are like the main glaring obvious differences in the structure of RNA versus DNA. Before moving on, let's do a quick review. What is Chargaff's rule? It's the same as my rule. It's just more like science-y sounding. In any species, humans, me, you, plant, potato, the amount of A always equals the amount of T, and the amount of C always equals the amount of G in DNA. What comprises the backbone of DNA? Think of my biology rock band name. Sugar phosphate backbone. Yeah, sugar phosphate groups. Sugar phosphate groups. That's the, it's got the pentose sugar over here with that phosphate. Nitrogenous base over here. Yep, sugar phosphate groups. You'll see that again too. This is going to be important when we're building our own DNA. So this is something that we're going to see physically in our hands, which is cool. Which nucleotides are purines? If it's pure, then you're going to put a ring on it. Purines. Adenine and guanine are purines, right? So one from the AT grouping and one from the CG grouping. Again, that's just rote memorization. I'm sure we'll figure out a way, like a little acronym or analogy to remember those. What type of bond holds together nucleotides? Think about it. It's the bonds that together we are strong, and one at a time we are not. They are hydrogen bonds. Quick review continued. Summarize the scientific contributions of Chargaff, Franklin, Watson, and Crick. This one's not really College Board specific. This is just knowing the history. So Chargaff did the A to T to C to G. Franklin did the helix shape of the DNA. And Watson and Crick kind of pulled it all together. Watson and Crick get all of the recognition for this stuff. Franklin and Chargaff are just kind of like left in the dust. What are some key differences between RNA and DNA? This one you do have to know for College Board stuff. DNA double-stranded, A bonds with T, RNA single-stranded, A bonds with U. Also deoxyribose and ribose. Practice free response question. I'm not going to give you the answer to this one. You're going to have to do this on your own. I'll check it when I check your notes. Let's read. Actually, hold on, let's make sure. Yep, no answer. So this one we'll go over together. Many species of bacteria are becoming resistant to antibiotics. No bueno. Recently, studies have analyzed the chromosomal DNA of, I thought I had my little thing. Chromosomal DNA of antibiotic-resistant bacteria. One bacteria studied is methicillin-resistant Staphylococcus aureus. We know that as MRSA. Researchers have found that the chromosomal DNA does not contain the gene for antibiotic resistance. Weird. They also found that when MRSA is cultured with a non-resistant bacteria, then the non-resistant bacteria becomes antibiotic resistant. It kind of sounds like a horror movie. You, as a scientist, I'd like you to identify the location of the antibiotic resistant gene. And discuss how the antibiotic resistant genes are being transmitted to non resistant bacteria when they're cultured together. Amazing. Thank you for doing science with me. I'll see you in class.